Today's video is about DNA replication. DNA is one of the most significant parts of biology. It contains the genetic code for all living things. DNA replication is the basis of biological inheritance. DNA has a sugar phosphate backbone, which is made up of phosphates, the yellow circle, and deoxyriboses, the orange pentagon. These two are bonded together in a long chain. Here we have a small section of DNA which has already been unwound from its typical double helix structure by the enzyme topoisomerase. Then helicase, the unzipping enzyme, comes along and breaks apart the hydrogen bonds between the DNA bases, adenine, guanine, thymine, and cytosine. Once helicase has broken apart the bases, another enzyme called primase comes in and creates an RNA primer, the blue pentagon, to let the DNA polymerase know where to start building the new DNA. Once the primers have been laid, DNA polymerase begins building new bases. DNA is typically read from 5' where the phosphate is to 3' where the deoxyribose is. This is why the top strand, known as the leading strand, and the bottom strand, known as the lagging strand, builds in opposite directions. Because DNA must be built from 5' to 3', the lagging strand has to wait until the helicase has cut through enough bases for the primase to jump up, giving the DNA polymerase a signal as to where to build new bases. Once helicase has reached the end of the DNA sequence being replicated, and the DNA polymerase has built the new complementary strands, an enzyme known as ligase comes in to glue together the sequences of DNA on the lagging strand. Once the ligase has done its job, we now have a second copy of the DNA we originally began with. This means that our DNA has been successfully replicated.